lastly in resistance to pathogenic insects, and later on we started discussing about uh, physiological mechanisms of resistance. And in the physiological mechanisms of resistance, first we discussed about the increase in thickness of or reduced penetration of insecticides through cuticle or gut. Second, what was discussed in one of the major factor which we were discussing about was with respect to detoxification of insecticides. We didn't complete it. And the next factor also today we will try to discuss, that is target site insensitivity. And one more thing, physiological mechanism is remaining, that is active excretion by ATP binding gases. So in the increased detoxification of insecticides, we discussed about esterases and glutathione transferases. Now, today we will be discussing about two more enzyme systems. Just a minute, I will share it. The first one will be mixed function oxidases or cytochrome P450 monooxygenases. Can you see the presentation?
Yes, sir.
Okay, fine. So, actually, it's a large group of enzymes, not a single enzyme. Glutathione is transferred, it's a single enzyme. But mixed function oxidase diverse functions, and most of it is oxidative in nature. And mostly it have cofactor cytochrome P450. That's why it is also called a cytochrome P450 monooxygenases. And it has the ability to, if we say many papers, actually, I forgot the name of the scientist who work, and actually that scientist is in Cornell. Scott, yes, Jeffrey Scott. Jeffrey Scott is one scientist who works extensively on cytochrome P450 monooxygenases. So, in one of his paper, he has written, and it is a group of enzymes which has genetic diversity. That means it is genetically determined by a diverse array of genes, not a single gene is contributing to the formation of this enzyme. And it has broad substrate specificity. That means it is not that specific to a particular molecule. It can react with a diverse kind of molecules. So that is one second character, that is broad substrate specificity. And the third character is nothing but it has the ability to catalyze different kinds of reactions. Though I told about oxidative reactions, it can also catalyze other kind of reactions. So that is the beauty of mixed function oxidases. That's why it is also called as mixed function, because of its genetic diversity. That means a number of genes. I will discuss about there is the number of genes in the next slide. Genes can lead to the synthesis of this group of enzymes. It can react with a variety of substrates. So, take the case of esterases. We'll compare it with esterases. Do you remember yesterday when you told what kind of molecules are hydrolyzed by esterases?
Amides,
No,
esterborne
what kind of
amide,
molecules? No,
pyrethroids,
ester,
phos. Hmm.
yes, ester, amide, and thioester. Any molecule with an ester, thioester, or amide bond. So that is because it's an esterase. It has to have this kind of ester bonds or something to break hydrolyze upon. But mixed function oxidases, it doesn't have that much specificity. It is broad in action. That's why it is called as broad substrate specificity and catalytic versatility. Not only oxidation, hydrolysis, everything can be carried out by this group of enzyme. And another important feature with the case of mixed function oxidases is, though we are discussing in the case of insecticide resistance, mostly we discuss in the case of degradation reactions. That means detoxification reaction. But mixed function oxidation is also involved in activation reactions. Have you heard about pro-insecticides? What is a pro-insecticide?
They are inactive in
Inactive, not inactive. It is active.
uh, so for example, hello.
Yes.
So in case of OP compound,
Mm.
um, so many insecticides required. If they are not active initially when we are applying, while applying they will get active.
Partially it is correct. I will correct your definition in a minute. But it gets converted into a more active form in the biological system. That is a pro-insecticide. So it's yeah. So that is one thing. It is an active compound. Take say I will take the case of example of malathion. Malathion. Malathion is a pro-insecticide. Malathion as such molecule is having some toxicity. But in the insect system, because of the activity of mixed function oxidases, this malathion will get converted into more toxic malaoxone. So for this conversion to malathion to malaoxone, to make the insecticide more toxic, we need the enzyme mixed function oxidases. So that is one critical feature of mixed function oxidase. Earlier we talked about esterases and glutathione transferases. Both are uh, involved in degradation reactions or metabolism only. Here it is not so, it is involved in activation reactions also. And another feature is because of its broad substrate specificity, that means it can react with a diverse kind of molecules. It's involved in the metabolism of organochlorines, organophosphates, carbamates, synthetic pyrethroids, insect growth regulators, whatever be the pesticide molecule. It can react with them and it can degrade. That is because of its broad substrate specificity. And mainly it is due to the, as already told, upregulation, that is more production of the enzyme with the existing gene set, or the insect will make more copies of the gene amplification. That's the case with other enzymes also. And another character is that it is one of the largest families of enzyme, and the major function of it is to convert lipophilic one or hydrophobic molecules into hydrophilic ones. Yesterday we have discussed in the case of what is the why a molecule has to be converted into hydrophilic one or water soluble one. Yesterday we discussed in the case of glutathione transferase.
To excrete.
Yeah, it can be easily excreted. Yes. So the next one, numerous substrates and all. So because of all these characters, this group of enzymes, mixed function oxidases, is also called as diversisozymes. And there are, I told you, there are different number of genes which express different kinds of enzymes. So we have different isoforms. So it is given different names. I see the last point. See, there are more than 100 genes in this family. These 100 genes can lead to the formation of a different kind of enzyme. So they have given a kind of nomenclature wherein any of this enzyme group will be given with the first name will be CYP because of the cytochrome P450. That's the one. So CYP will be there. That is for all this group of enzymes. So CYP is common, and in the next it will be the family number. and this family number is given based on the amino acid sequence. If two enzymes has 40% amino acid similarity, so we have one cytochrome P450 enzyme, say A, we have cytochrome P450 another enzyme B. If both these two enzymes are having 40% amino acid similarity, both will be categorized into one family. So that will be given one, each family will be given a number. And there are more than 25 different families of cytochrome P450 enzyme. That means more than 100 P450 genes are there, and they all these genes will lead to the production of these enzymes, and these enzymes fall into 25 different families because these enzymes share similarities in amino acid sequence. So if two enzymes share 40% amino acid similarity, it will fall in one family. So we have 25 different families. And the second thing, so you can see this one example I have given is CYP6A1. The 6 CYP, that is cytochrome P450, that is common for all this group of enzymes. 6 is the family number, and A, this is the subfamily name. And this subfamily, within the family, those enzymes which have 55% amino acid homology, which share similarity of 55% amino acids, they are given a, a name, a common name. So that is the second one, CYP6A. A is those having they denotes the subfamily, and those enzymes within that subfamily share 66% amino acid similarity. And the last one is the gene. Once you come to last gene, it will differ. So we have 100 different genes in 25 different families. So this is how, because I have given this because if you scan through literature, you will come across this kind of nomenclature of cytochrome P450, especially if you go through that Jeffrey Scott paper, you will see a lot of this nomenclature. Are you clear with cytochrome P450?
Sir,
Ah,
sir, I have small
Last
doubt.
number
ah,
one.
yes.
So
Uh,
CYP 6, they mentioned as 6, right? So 6
ah,
means they're having 46% human acid homology.
40% amino acid homology.
So then what's the reason behind why they kept 6, number 6 only?
Number six only because there are different. See, there are 25 different families. They numbered
Yeah.
it from 1 to 25.
Okay.
I have taken only one example. I have taken SIP6A1 because this is one of the I've taken. I randomly chose SIP6A1 because only because this is one of the largest group of cytochrome P450 gene which contributes to insecticide resistance. That's why it took SIP6A1. We can have SIP1, SIP2, SIP3, SIP4, SIP5 up to 25.
Okay.
Uh, and in SIP 1 itself, we can have SIP 1A, SIP 1B, SIP 1C, SIP 16, 1C, D, E, etc. Understood?
Okay, sir.
Safna,
Yes, sir.
what you asking?
Last, not the gene number. Yes, sir.
Yes, it's not actually the gene. Actually, it is, you say in the same family, in the subfamily itself, there will be they differ in their genetic makeup. That's the thing. See, it's one, two, three, all those things will be given usually, especially in that last, the gene. Whichever gene is discovered first, it will be given one. The second one will be two. Just like we name the mutations or in coronavirus, isn't it?
Yes,
1.1,
sir.
1.2, 1 point, till we have doesn't have any mutation, it will be given 1. If we have mutation to that particular gene, it will be given 1.2. If there another mutation happens, we will give 1.2, isn't it?
Yes, sir.
Now you are saying about that delta, a variant of that delta strain. So it is given, I think, zero point 0.1 or zero point 0.2 or something, I forgot. So that similar way only it is named. Okay. Now, usually whenever we discuss about detoxification enzyme, we usually discuss about these three only: esterases, glutathionase transferases, and cytochrome P450 monooxygenases. And now we have a new group of enzyme which is more and more gaining importance in insecticide resistance. Say after 2012, there are a number of papers which suggest uridine diphosphate glycosyl transferase as imparting resistance. This is an enzyme which helps in the transfer of glycosyl group to pesticide. So once a glycosyl group from sugar molecule, from once this glycosyl group is transferred to pesticide, that pesticide will become hydrophilic and it can be easily excreted because sugar is one of the most hydrophilic compound, so it gets easily water soluble. So if you scan through little, though this UGT enzyme, uridine diphosphate glycosyl transferase enzyme, was reported in living system for long, but its role in insecticide resistance was studied thoroughly only lately, and mainly after the uh, development of this molecular biology, especially this expression studies, mRNA expression studies, and all, after real-time PCR and all became extremely popular. Now, this more large number of papers are coming up where this enzyme, uridine diphosphate glycosyl transferase, is attributed for resistance to most of the new molecules of enzyme. So the role of this enzyme is to catalyze the transfer of glycosyl group from an activated sugar. Activated sugar means UDP glucose. UDP is uridine diphosphate. Diphosphate, just like ATP, adenosine triphosphate, is an energy containing compound. So this UDP will bind with glucose, and this activated sugar, once glucose binds with UDP, this gets activated, becomes more energetic, and this glucose moiety, this from this activated sugar will be transferred to xenobiotics or pesticide molecules. So once this glucose is transferred to this pesticide molecule, the pesticide molecule will become more water soluble and it can be easily excreted. And C, it is main involved in the detoxification of many of the pesticides, especially with respect to new molecules, neonicotinoids, diamides. I will take two examples. Can you hear me? Can you hear me?
Yes, sir.
Okay, I think I lost network connection. So it is implicated in resistant neonicotinoids, diamides, this lipid biosynthesis inhibitor molecules, spirotetramide. You might have heard about spiromesifen. There is another pesticide belong to the same group, that is pyrotetramat. Pyrotetramat, one different, almost it is same as spirometz when effective against sucking pests as well as mites. And I think pyrotetramat is having systemic property also. So that is one difference. So I will take two examples
Sir,
from the
voice breakdown.
voice is breaking.
കേൾക്കാൻ പറ്റുന്നുണ്ട്.
കേൾക്കാൻ പറ്റുന്നില്ല. Just a minute, just I will check.
ഇടയ്ക്ക് കട്ടാവുന്നുണ്ട്.
There was a message that my network connectivity is low, so that's why I checked in between. Let me check my network. It's a bit low. Okay, one thing, I will switch off my video. Okay, can you hear it now? Is it clear?
Yes, sir.
Yes, okay. So here are so for first paper, I not given the authors. Actually, what they did was they studied the expression of UGT genes. Three genes you can see: UGT 344B4, UGT 344C7, UGT 344N4, and they studied the expression of these three UGT genes in Aphids gossypi. From this is from BT cotton. They collected this aphid from BT cotton, and they studied their susceptibility to imidacloprid. This first one, this first bar, this is the imidacloprid susceptible aphid they maintained in their lab, and these three are resistant population or field population they have collected. See the expression level. The gene expression is extremely high in population compared to the susceptible one. So that means that the expression of gene is more and the production of uridine diphosphate glycosyl transferase enzyme is more, and it is leading to the detoxification of imidacloprid and imparting resistance to imidacloprid. That is with respect to imidacloprid. There are a lot of examples, papers with respect to resistance to neonicotinoids by this UGT gene expression. And this is with respect to chlorantraniliprole against striped stem borer Chylosaturasalis in rice. See this. Uh, Here in the x-axis there is given numerous UGT gene, and they are taken susceptible one and resistant one. See the expression level in the resistant expression of this particular gene in the resistant one. Extremely high level of expression of gene. That means extremely high level of UGT enzyme, again contributing to insecticide resistance. Is it clear?
Yes, sir.
Yes. Okay, fine. Then coming to the next one, so we are completing the increased detoxification. First, we have seen decreased penetration. Second, we have seen detoxification. Glutathionase transferases and then UGT. And coming to the next mechanism, that is target site insensitivity. Actually, target site insensitivity is as important as this increased detoxification. So what will happen is that the target site will become insensitive to the insecticide. So I'll ask you a question: which is the target site for neonicotinoid?
Acetylcholine.
Acetylcholine. Can you name it? You are in PhD, so can you name specifically which kind of
Acetylcholine.
acetylcholine? Which kind of acetylcholine?
Nicotinic, nicotinic acetylcholine.
Nicotinic acetylcholine receptor. Okay, okay, fine. So, if the nicotinic acetylcholine becomes insensitive to neonicotinoid, that means it doesn't sense neonicotinoid. What will happen? It doesn't respond to neonicotinoid. Doesn't its response is binding. It doesn't if it doesn't bind to neonicotinoid, what will happen?
Bind to some other molecules which are similar to.
Yes, I couldn't get you Bindu properly. What you were telling.
So it will bind to the same group which mimics acetylcholinesterase.
Okay, which mimic acetylcholine. Okay,
Mm.
okay, fine. But whether it will bind to neonicotinoids?
No, sir.
No, because it has become insensitive to neonicotinoids. So how that insensitivity happens? We told you about we told you in earlier that all these receptors, nicotinic acid, whatever be the receptor molecules, they are proteins. And any molecule to bind to that protein, we discussed about lock and key hypothesis. They have to perfectly match. So if a mutation happens in that target protein, the receptor protein, mutation means an amino acid changes, and once that amino acid changes, the structure of the protein also changes. But the change in structure makes the protein, receptor protein, insensitive to or not responsive to pesticide molecule. Still, that receptor maintains its sensitivity towards the original neurotransmitter. Here it is acetylcholine. I will repeat. There is a target protein or receptor protein. Here we are discussing about neonicotinoid. So the receptor protein is nicotinic acetylcholine receptor. A mutation occurs in that nicotinic acetylcholine receptor. So there will be a replacement of one amino acid with another amino acid. So once the amino acid changes, the structure of the receptor changes. The structure of the nicotinic acetylcholine receptor changes. So if it changes drastically, it won't be able to bind to both insecticide as well as its neurotransmitter. The neurotransmitter, its ligand here is neurotransmitter or ligand here is acetylcholine. If acetylcholine also can't bind to the nicotinic acetylcholine receptor, near transmission won't happen. So what will happen is that it changes the structure, but it will maintain its affinity towards the ligand, natural ligand. Here it's acetylcholine, and it loses its sensitivity towards the pesticide. Understood?
Yes, sir.
Okay. Just like
Yes, sir.
just like a lock and key.
Voice ഇടയ്ക്ക് break ആയി.
വോയിസ് ഇടയ്ക്ക് ബ്രേക്ക് ആവുന്നുണ്ടോ എപ്പോഴും? ഹലോ,
ഇല്ല, ഇപ്പൊ കുഴപ്പമില്ല.
ഇച്ചിരി നെറ്റ്വർക്ക് ഇച്ചിരി മോശമാണ് ഇന്ന് എന്താണോ എന്തോ. So one thing we have to keep in mind is that we are discussing about lock and key. This lock and the key has having several grooves, and a slight change in the groove of the lock makes one key unsuitable to that lock while making the other key still able to open that lock. So that's how it works. So this mainly happens with the help of point mutation. I told discussed that triplet codon which codes for each amino acid. I shared that thing. So if one point mutation occurs, it will lead to the change in the protein structure of the receptor molecule. And target site insensitivity has different mechanism. One such mechanism is decreased penetration through nerve sheath. That means the insecticide won't be able to penetrate through the nerve sheath. Some mutation occurring makes the nerve sheath impermeable to the pesticide. Then mutation in ligand gated ion channels, mutations in voltage gated ion channels, then mutations in acetylcholinesterase gene. So that means OP and carbamate, they bind to acetylcholinesterase enzyme. So if the acetylcholinesterase gene, acetylcholinesterase enzyme makes a slight conformational change, which makes it insensitive to OP and carbamates, but still maintain its sensitivity towards acetylcholine. So that can happen. And mutation in chitin synthase gene, mainly with respect to chitin synthesis inhibitor enzyme, then mutations in mitochondrial gene, those insecticides which act on respiration, cellular respiration. Okay, we'll see it one by one. The first one is decreased penetration through nerve sheath. This is mainly an old mechanism. This mechanism is not that studied nowadays. I don't know whether any papers are there with new papers are there with respect to this aspect, decreased penetration. All the literature cites this old study wherein there is a decreased penetration of organochlorine insecticides, DDT, dieldrin, and permethrin, synthetic pyrethroid, in resistant housefly musca due to a gene named PEN. PEN is the gene name. There is a mutation in the PEN gene, and with this mutation in this PEN gene is the one which helps in the penetration of this DDT, dieldrin, and permethrin into the nervous system. But because of mutation in this PEN gene, which helps in the penetration, it prevents the penetration of this enzyme, and this houseflies become resistant to DDT, dieldrin, and permethrin. So that is decreased penetration through nerve sheath. Coming to the next one, that is mutation in ligand gated ion channel. I think you still remember what is a ligand one, what is a ligand gated ion channel. So here you can see the major neurotransmitters or ligand which you have studied with respect to pesticides is acetylcholine, gamma aminobutyric acid, and glutamate. So these mutations can happen on receptors of this ligand, nicotinic acetylcholine receptors, GABA receptors, or glutamate receptors. So this mutation can happen on ligand-gated ion channels, mainly with respect to nicotinic acetylcholine receptors, GABA-gated chloride channels, and glutamate receptors. Here, I am taking only a few examples. I'm taking only a few examples. If you see literature, each and every study points to different mutation. With each insect, there can be a slight mutation. I am here taking only a few examples. Don't think that in nicotinic acetylcholine receptor, only the mutation which I have mentioned in the slide occurs. No. In different insects, different kinds of mutations occur. This is only a few mutations which are common in insects, and there are several other mutations in nicotinic acetylcholine receptors which leads to resistance. And even with different kind of insecticides, again the mutations vary. So in nicotinic acetylcholine receptors, single point mutation. Here mostly in many of the insects what we are seeing is at the 151st position. Tyrosine was there originally in the original nicotinic acetylcholine receptor. If you compare the susceptible one and the resistant one, in the susceptible one at the 151st position of this gene we have tyrosine, but in the resistant one it is replaced with serine. And in the in the 81st position, earlier it was arginine in the susceptible one. In the resistant one, a single point mutation led to the formation of threonine. The amino acid became threonine instead of arginine. And this conformational change makes the nicotinic acetylcholine receptor resistant to insecticide. Clear?
Yes, sir.
Yesterday, I have shared a few two pictures with respect to amino acids. So one thing you check it with tyrosine and serine. See the triplet codon for tyrosine. I will just check in that tyrosine and serine. Okay. The triplet codon for serine is UCU, uracil, cytosine, uracil. And for tyrosine, it is UAU. Serine, it is UCU. Tyrosine, it is UAU. So one replacing adenine with just a minute, adenine with cytosine is the only thing which the insect has to do to become resistant to insecticide. So that is single point mutation, wherein that mutation leads to the development of a new triplet codon. A slight change, only one codon is changed. Then the insect becomes resistant. So that is how insects can develop resistance pretty quickly. A single mutation is sufficient. It's not a bigger thing for insect.
Sir, but a mutation favorable to the acetylcholine
And
ആവുന്നുണ്ട്.
coming to the
Acetylcholine.
Couldn't get you properly. Hello.
സർ ആ മ്യൂട്ടേഷൻ നമ്മുടെ അസെറ്റൈൽ കോളൈന് ഫേവറബിളും കൂടെ ആവണ്ടേ നടന്ന മ്യൂട്ടേഷൻ
Acetylcholine.
അസെറ്റൈൽ
Hello.
കോളൈന് അവിടെ ബൈൻഡ് ചെയ്യാൻ കഴിയണ്ടേ അങ്ങനെയുള്ള മ്യൂട്ടേഷൻ ആവണം എന്നല്ലേ
Yes,
പറയുന്നത്.
that's what I told you. This mutation will make the receptor insensitive to insecticide, but it will remain sensitive to the ligand, the natural ligand, that is acetylcholine.
ഓക്കെ.
Understood? That's what I told. In target site insensitivity, in all those cases, it will be the same. This conformational change makes the receptor insensitive to the pesticide, but it will remain sensitive to the ligand, natural ligand. Here it is acetylcholine. In the case of GABA, it will be GABA. The natural ligand is GABA. So here one of the most common gene which got mutated because of GABA resistance is RDL. RDL is nothing but resistance to dieldrin, because in a Drosophila strain which became resistant to dieldrin, uh, what they found was there is a mutation in the 302 position. In the 302 position of the gene, GABA gene, actually earlier it was alanine, and in the resistant gene, it got converted to serine or glycine. And those GABA genes with mutated serine or glycine at the 302 position, though that GABA gene was called as RDL gene, resistant to dieldrin gene. So in the GABA gated chloride channel at the 302 position, in the susceptible insect it was alanine, but in the resistant position insects it got converted to serine or glycine. And those the individuals with this kind of mutation, that gene is called as resistant to dieldrin or RDL gene. And this A302S mutation, that means at the 302 position is alanine to serine point mutation, confers resistance not only to cyclodienes but also to fipronil. So it will lead to what kind of resistance? Multiple resistance or cross resistance?
cross resistance.
One answer is multiple, one answer is cross.
So both
One
are
thing is
belong
that
to similar mode of action, right?
yes, it is similar. If you consider mode of action, it is cross resistance only. If you consider different group, it will be multiple resistance. That is why I asked, specifically asked. So you answer it in that way because it is mainly according to Iraq classification. It should be cross resistance only because they have that classification is based on mode of action. If it is in the traditional chemical classification, it will be multiple resistance. So that's you remember. So it can be confusing. That's why I specifically asked this. And if in the case of resistance to fipronil, we have few more mutations which are detected at the 301 position alanine to glycine again, at the 20th position alanine to aspartic acid, and at 350th position 309 to methionine. I don't want you to remember all these mutations, at least one or two mutations. If you remember it, it's more than fine. Just to give you a glimpse that there can be different kind of mutations which can contribute to resistance. Okay. Coming to the next one, that is mutations on glutamate. Again, we are discussing about ligand gated mutations in ligand gated chloride channels. The next is glutamate gated chloride channels. These mutations confer resistance to avermectins. And abamectin is specifically an acaricide. It is also have an insecticidal property. And scientists have discovered that at 320th position of this glutamate chloride channel gene, in susceptible individuals it was glycine, and in resistant insect it is aspartic acid. D is aspartic acid. I think D is yes aspartic acid. So if the mutation happens from glycine to aspartic acid at 323 position, the insect will become the mite will become resistant to abamectin. And in diamondback moth, this mutation from proline to serine at the 299th position confer resistance to ivermectin. So that is mutations in glutamate gated chloride channels. So first we have seen decreased penetration through nerve sheath. The next we have seen mutation in ligand gated ion channel. Now we will see mutations in voltage gated ion channel. And this voltage gated ion channel also we have membrane proteins which permits the movement of ions across the membrane. So any mutation which is happening to that ion gated voltage gated ion channels, it change to a change in the membrane voltage. Mainly this mechanism we are seeing in the case of two ion channels. One is sodium channel and the other one is calcium channel. So sodium channels is the one which is mostly studied because pyrethroids are acting on sodium channels, and many of the mosquitoes have developed resistance to pyrethroids. So what they found that mutations in this sodium channels, voltage gated sodium channel, all voltage gated sodium channels are proteins. So that mutation reduces the binding affinity of insecticides to this ion channel. So those ion channel which has lost its affinity towards insecticides or specifically pyrethroids, those kind of sodium channel gene, voltage gated sodium channel genes are called as KDR gene. Remember this KDR gene. KDR is knockdown resistance. Pyrethroid, one of the common mode of action of pyrethroids is knockdown resistance. So knockdown action, not knockdown resistance, knockdown action. That's why if there is a mutation happening in the sodium channel gene which confers resistance to pyrethroid, that gene is called as knockdown resistance. And in knockdown resistance, what they mainly observed was uh, two point mutation. First point mutation they observed was in the 1014 position, where it was leucine in the susceptible insect. It got mutated to phenylalanine, histidine, or serine. So that was the first one they discovered. In a resistant insect, they found that instead of leucine at 1014 position, either it was phenylalanine, histidine, or serine. But later on, in certain other group of insect resistant insect, they found that along with the mutation at 1014 position from leucine to phenylalanine, they also detected methionine to threonine point mutation at 918 position. So if an resistant insect is having both these mutations at 1014 position and 918 position, those individuals with that kind of gene, these 2-point mutations, they were called as super KDR. So those individuals with a single point mutation at 1014 position, those kind of genes were called as KDR. And if the gene is having 2-point mutation, one at 1014 position and another at 918 position, they uh, they were called a super KDR. This was called very early, maybe in the 90s and all. And later on, with the advances in molecular biology, now they have identified more than 26-point mutations conferring resistance to pyrethroids in voltage gated sodium channels. So it's not only these two. Earlier they detected only two. That's why they called it as KDR and super KDR. Later on, they identified so many different kinds of point mutations. Okay. Next one is mutations to calcium channels. What can be the insecticide which to which insects has evolved resistance if they got mutations in calcium channel? What group of insecticide? Which are the insecticides acting on calcium channels? Anyone? I have written in that slide also. Which are the group of insecticides acting on calcium channels? A mutation in calcium channel affects which efficacy of which group of insecticide? Hmm? I'll have to conduct a few exams, then only you will refer the older notes. Aren't you unable to hear me?
No, sir. You can hear me.
Then, which is the group of insecticides acting on calcium channels? Diamides, I have written in the slide also. It is chlorantraniliprole and flubendiamide. See, in the case of diamide resistance, they mainly the first report of diamide resistance was the field report was from diamondback moth, and what they observed was in diamondback moth at the 4946 position. Earlier it was glycine, it got mutated to glutamic acid. Okay, fine. They observed this mutation from one location. I think it is from US or something, and this same point mutation, they observed it from another location for contributing to resistance to chlorantraniliprole. So that means diamondback moth in two different geographical locations. I think that is these two locations are in two different continents. I forgot that exact locations. I have that paper, but I forgot that exact location. Actually, these two locations are from two different continents itself. Even if these insects are geographically isolated, the mutations that is happening to diamide insecticides is the same. That means wherever the insect is, if they are exposed to a particular kind of insecticide, the insect groups, even if they are separated, they have the ability to develop resistance in the same manner, the same kind of mutation. So that is with respect to mutations in calcium channels. Coming to the next one, that is mutations in acetylcholinesterase gene. Insects mostly they have um, uh, two genes, that is ACE1 and ACE2. These are the acetylcholinesterase gene. That means this gene will lead to the synthesis of acetylcholinesterase enzyme. These ACE1 and ACE2 genes will lead to the synthesis of, are responsible for the synthesis of acetylcholinesterase enzyme. If there is a mutation happening in this enzyme, this enzyme acetylcholinesterase will become insensitive to acetylcholine, no, insensitive to opium carbamates while retaining its sensitivity towards acetylcholine. So opium carbamates, it has to bind to acetylcholinesterase enzyme to inhibit the activity. But slight mutation occurred in this acetylcholinesterase enzyme, and the insecticides, opium carbamate insecticide, won't be able to bind and won't be able to bound to this acetylcholinesterase enzyme, but it will be able to interact with acetylcholine. So that acetylcholine can be broken down into acetic acid and choline. So another thing that happens is in the mutation is gene duplication. There will be insect will make more copies of this gene. Once there are more copies, more production of acetylcholinesterase enzyme. So that means more production of acetylcholinesterase enzyme means we have to use more quantity of the pesticide to inhibit the enzyme. So that is also happening. So the common mutation that is observed is at the 119th position from glycine to serine. At 119th position, glycine is there in the susceptible group, while in the resistant group, it got mutated to serine. And once that mutation happens, this acetylcholinesterase enzyme will become insensitive to opium carbamate group of pesticides. Coming to the last one in the case of target site insensitivity, that is in the case of chitin. Okay, one more is the second last one. So the mutation in the chitin synthase gene. This is one report from the case of mite growth inhibitor, the etoxazole. Remember this etoxazole. This is a mite growth inhibitor. It inhibits chitin synthesis in mites. This mutation confers resistance to etoxazole in Tetranicus atticae. This mutation in chitin synthase gene. So in the next last one is mutation in the mitochondrial gene. Here it's reported in bifenacid. It's not only reported from bifenacid. There are so many reports. I've given only one. This mutation in cytochrome B gives resistance to bifenacid in Tetranicus atticae. So these mutations in ligand-gated ion channels, mutations in voltage-gated ion channels, mutations in mitochondrial gene, mutations in chitin synthase gene, all can of this can contribute resistance to insects. I will take excretion to ABC transporter. That is the last physiological mechanism tomorrow because it's already 1 o'clock. Any clarifications you need? If you go through literature now, they mostly study target site insensitivity along with traditional resistance mechanisms. So that is the latest trend because carrying out a genetic analysis is pretty easy now. Sequencing of taking out a gene and sequencing that gene, it's pretty easy now with the development of sequencing techniques. Even maybe after 5 years, within 5 years, we will be taking out the entire whole genome of a susceptible insect. We'll take out the whole genome of resistant insect. We'll completely sequence it, and we'll find out the difference in the genetic makeup. Now we are targeting a specific portion of the genome. specific with respect to that particular gene coding for a particular kind of enzyme or a protein. But mm, later on, the genome sequencing genome is becoming extremely cheap now. So instead of going for targeting a specific gene, we can go for the entire genome. So that is one thing which can be done. Okay. Any clarifications do you need?
ലാസ്റ്റ് കൂടെ എക്സ്പ്ലെയിൻ ചെയ്തിരുന്നോ
Either?
ABC transporters?
No, no, no. ABC transporters I will take later because I will take about 5 to 10 minutes for ABC transporters.
ഓക്കെ കണക്ഷൻ ലോസ് ആയപ്പോൾ ഞാൻ വിചാരിച്ചത് എക്സ്പ്ലെയിൻ ചെയ്തു.
No, 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 no. One thing you can check in Moodle is Moodle. I have made it almost up to date by today morning. I have uploaded all my lectures, the PDF of all the PPT, reading materials, and a few classes I forgot to record. Other than those, all those I have recorded is uploaded. So if you want to refer it, you can refer it also. I will take up a small exam in Moodle also because you have to go through this. Otherwise, you will forget all those things. Okay, that will be a one-word exam. Don't think it is a big exam. Just to refresh you whatever you have learned in the first from the first book, the iron channels, sodium channels, potassium channels, etc. Next week I will conduct. Okay. Tomorrow again at 12 o'clock, isn't it?
Tomorrow, classes, I will inform you.
Okay, okay, fine. Thank you.
Thank you, sir.
Thank you.